Conference and our discussion with NerveGen, a company working in spinal cord injury and making some really interesting progress on behalf of patients with super high unmet need. It's a pleasure to introduce Mike Kelly, President and CEO. Thank you, Mike, for joining us here today.
Thanks for having me, Jay.
All right, so maybe for those in our audience who may not be as familiar with the NerveGen story, can you just give us a brief overview of NerveGen and what you're currently working on?
Sure. So we're a clinical stage biotech company, an emerging clinical stage biotech company, I should say. We are based in Vancouver, public on the TSXV, and we have two programs. So our first program, NVG-291, as you mentioned, is in spinal cord injury, and we are having a phase 1b/2a data readout next quarter. And our next program, NVG-300, is a little bit earlier stage, and we'll have a readout this quarter in the preclinical models for ALS and stroke. So the company is working on neuroreparative therapies.
Okay, great. And maybe also, can you just provide some background about yourself and how you chose to join NerveGen?
Sure. I've been in the pharmaceutical industry for 30 years, started out on the commercial side of the business with big company like TAP Pharmaceuticals. And then I jumped out into the world of startup with companies like Guilford and Vira Pharma, and then got to pure startups, which are Azure and Adapt in the spec pharma space. But over the last couple of jobs that I've had, I worked at the C-suite level at a company called Covis, as well as my last job. I worked with a group of the founders for a company called Adapt, where we launched Narcan nasal spray in US and Canada. That was a very gratifying job, which led me to NerveGen. Actually, why NerveGen? The technology, the people, the board, and the opportunity to help a lot of people. I met Jerry Silver early on in the interview process, and Jerry recently passed, but Jerry's a pioneer in this space. He's a scientist who discovered our lead molecule, NVG-291. He's been working in spinal cord injury. He worked in spinal cord injury his entire career, and he was a great teacher. So he got me very excited about this technology and how it can apply to multiple different diseases that have significant unmet medical need. Then I met the board, I met the management team, and I felt that, you know, I can help this company move forward pretty rapidly and get to commercialization.
All right, thank you. That's a super impressive background, and I had the pleasure of speaking to Jerry. So,
yeah, he's a great gentleman.
Maybe just to dig in a little bit more in detail on NVG-291, can you talk about how it was discovered and how it works?
Sure. Actually, Mark DePaul could probably explain a lot better. He's actually on our staff here at NerveGen, but he worked in Jerry's lab and he was on the team that actually discovered it. But Jerry made a series of discoveries throughout his career, but several of them led to understanding why nerves don't repair when there's a traumatic event like a spinal cord injury. And they're all centered around a group of molecules called CSPGs, chondroitin sulfate proteoglycans. So they are the molecules that basically protect and hold your central nervous system intact. They're the cement that holds everything together. But when there's damage, they are actually they collect in the glial scar that is formed around the injury, and they inhibit repair. You know, they inhibit regeneration, remyelination, and plasticity. And what Jerry did after making that discovery was the team started to look at ways to stop those inhibitors from inhibiting. And that's what led to NVG-291. They designed it after the intracellular wedge domain of a receptor that the CSPGs attach to and disrupt the signaling into the cell. So we know all of the biological processes that are stopped or that are actually turned back on because we're inhibiting the inhibitor. So it has been demonstrated, and Jerry's demonstrated this pretty broadly in a lot of his publications, that once NVG-291 enters into that process, nerves can actually regenerate again. so the body's natural mechanisms can take back over. You can remyelinate, you can regenerate, and you can enhance plasticity. We are now taking it to the next level and doing a lot of work to understand the molecular mechanisms that are what those specific signals are that we're turning back on.
Okay, great. And as we look ahead to your first human proof of concept study results, the phase 1b/2a study in spinal cord injury, can you maybe provide some background around the market opportunity for spinal cord injury?
Yeah, it's a pretty large market. There's, in the United States, there's roughly 18,000 cases per year of spinal cord injury. There's a group of 300,000 people living in the U.S. With spinal cord injury. And when we talk to patients, payers, and physicians, there's really nothing for these folks. They in an instant they have an injury, they're hospitalized, they have surgery to decompress the injury, and then they're on the rehab path, right? They do inpatient rehab, outpatient rehab, but there's nothing to help regenerate nerves. And they do get better to a certain extent, and they plateau right around 9 to 12 months, and then that's kind of where they are for the rest of their lives. So if we can actually show that we can turn back on the repair and make those connections and give some function back. We talked to payers, and payers said that they would treat us as an orphan type of drug. And if you take orphan type medicines and look at the prices that they're garnering in the marketplace, multiplied by the incidence and prevalence numbers I gave you, it's a multi-billion-dollar market opportunity. In fact, each case costs this system in the U.S. between $1 and $4 million over the course of the life of the patient. And, obviously it's a broad range, but it depends on the severity of the injury and the age that the injury was occurred.
Okay. And then can you walk us through the trial design?
Yeah, so we're studying two main things here. The primary endpoint is to measure connectivity, right? So we've chosen Shirley Ryan Ability Labs as our partner. Monica Perez specifically, she's the PI, and Monica is an expert in measuring that connection. She's an expert in electrophysiology. So we're actually measuring the electric current going from the brain past the site of injury to sensors on the hand and the leg. So the patients need to have a baseline measurement to get into the study, and then they're stratified into drug or placebo, and they go through an exercise routine, and we're measuring the improvement in the strength of that signal over the course of 12 weeks. So it's a 12-week treatment, daily subcutaneous injections, and then we're following for an additional 4 weeks, so it's out to 16 weeks, to see can we improve the strength of that signal, and it's called the MEP amplitude, or can we improve the speed of that signal? So that's the primary endpoint. And it's like a biomarker for spinal cord injury. It takes a lot of heterogeneity out of this patient population, which makes it very homogeneous, and it's a very objective endpoint. And then all of the secondary endpoints are functional. So they're like 10-minute walk time, 9-hole peg test, upper, lower extremity motor scores, pinch, grasp, all things that are very important to patients as far as functional improvement. So we're measuring that combination, improvement of signals and improvement of function. And the goal here is to show that we can improve signals, and that improvement in signals is actually resulting in some type of improved function.
Okay, great. So it sounds like a large opportunity. You have a well-designed trial. Anything you can share with us on the patient demographics that you've enrolled in terms of age, duration of disease, and nature of the initial injury?
Yeah, so there's actually two arms to this study, right? The first arm is what we're calling the chronic cohort. And that means people who've met that plateau that I talked about earlier. So their injury occurred between 1 and 10 years ago. And Dan Michael, our chief medical officer, presented some data earlier this year, I think on the first 10 to 15 patients. And it's roughly the mean time post-injury has been, right around 3 years, give or take. We won't know the final numbers until the rest of the cohort was fully enrolled. So the first cohort is the chronic cohort. And the average age is very typical to the average age of spinal cord injury. Early 40s, 43 is the average age of a spinal cord injury patient. In recent data, and this is mostly men. 70 plus percentage of the time it's men, and most of these injuries are occurring 70+% of the time due to a fall or some type of accident.
Okay. And then what is the natural course of the disease at, for example, at 3 years, have most of these patients plateaued in terms of their recovery?
Yeah, they have, and that's important because we don't expect to see any placebo effect here. Once folks hit that plateau, very few folks continue to improve. I'm not saying that all folks, there is a small percentage that continue to push themselves to make connections and try new different types of therapies or stimulation to continue to make and push those connections. But the majority of the population at that 9 to 12 months has plateaued, and there is very little improvement in motor skills after that.
Okay, understood. And you described a number of endpoints. Can you walk us through what, in your vision, successful outcomes would look like in terms of the primary endpoints and maybe some of the key secondary endpoints?
Yeah, absolutely. So the primary endpoint, the study was designed to meet the primary endpoint, which is the electric connections. So Dan worked with Monica and her team to look at actual MEP amplitudes and the changes in MEP amplitudes in some of the stimulation studies that they conducted. And they got to an N of 8 per arm. We bumped that up to an N of 10 per arm. So there's 20 people in this study to measure that primary endpoint, and we expect to see an improvement in connectivity. That's the first thing we'd like to see. The second thing we'd like to see is improvement in function. Obviously, when we meet with the FDA for an end of phase 2 meeting, we have to pick a functional endpoint moving forward, right? So we'd like to see some type of improvement in function. We have so many different secondary endpoints in this study, and the goal here is to take the primary endpoint, combine it with what we see in trends in the secondary endpoint, and just give us the ability to pick an endpoint for a larger study that we can conduct in phase 3. So, some of the examples of those would be, as I mentioned earlier, there's a score called GRASP, there's pinch force, there's 9-hole peg test, which is a progressive functional endpoint. There's 10-minute walk time for folks who can actually do a 10-meter walk test, or 10-meter walk test, not 10-minute walk time, 10-meter walk test. And then upper and lower extremity motor functions, so which are composite functions of the upper extremities and the lower extremities to see, is there any functional improvement over time? And we're measuring all of these endpoints at week 1 as a baseline, and then 2, 4, 8, and 12 to see, do these progress? So the goal here is to see statistical significance in the primary, and then separation in trends, strong trends in the secondary. That would be the first time ever in science that any company has ever proven that they can actually remake connections and turn those connections into some type of functional improvement. So we would be well onto phase 3 at that point.
Okay, well, that's an exciting landmark on the horizon to watch for. Can you give us some idea of the timing when we should expect to see that proof of concept data?
Yeah, we're telling folks, well, the last patient was entered the study December 31st of last year. It's a 16-week trial. And then we need, roughly 6 weeks to gather the data, clean the data, lock the database, and start our analysis. So we're looking, in Q2 in the May-June timeframe.
All right, we'll look forward to that. And then I guess as you look out on the horizon beyond that study readout, can you talk about when you could get your end of phase 2 meeting with the FDA, and maybe any initial thoughts on what a phase 3 study design could look like, especially in terms of number of patients